Hello everyone and welcome back to Clinical Cousins YouTube channel, where today we're going to go over how our brain interprets certain chemicals as taste. First, let's define some terms. Tastants are any molecules that have the quality of eliciting, bringing about taste as a sensation in our brain. These tastants bind to chemoreceptors that are in our taste buds. Now, these taste buds are not the little bumps on your tongue. These little bumps are called taste papillae. Anytime you see this word papillae, just know that we're talking about multiple little bumps. So it makes sense that the little bumps on our tongue are called taste papillae. So our sense of taste is called gustation, while ageusia is the lack of taste, which can happen when a person has a COVID-19 infection, for example. Hypogeusia is a decreased ability to taste. Hypergeusia is an increased ability to taste. These are sometimes called super tasters. And dysgeusia is an altered sense of taste. A great example of this is when a person is about to have a seizure, or they can have what's called an aura. So maybe they're tasting burnt toast even though there's nothing in their mouth. So we can remember the three types of gustatory cells by remembering taste buds. So what's really significant is the T, the S, and the B. So the T stands for the actual taste receptors. These taste receptors have little microvilli that act as chemoreceptors. Remember, chemical receptors that bind to and detect tastants that are on our tongue. These taste receptors are connected to primary afferent neurons. We also have basal cells. Now, these are the precursors to our taste receptors, and they will actually replace the taste receptors about every 10 days. So if you burn your tongue really bad, you've damaged the taste receptors, don't worry, because in 10 days, you'll have some more taste receptors which will fill in for that position. So lastly, we have the support cells, and at this time, it's not known exactly what the support cells do. Now, we have to clear something up. The bumps that we see on our tongue, they're not actually called taste buds. I have to say this again. They're actually called taste papillae. The taste buds themselves are, as you can see, are actually located within the taste papillae themselves. So we have three types of these papillae. We have the circumvallate, we have the foliate, and then we have the fungiform. Now, the circumvallate are the fattest, they're the largest, and they're located at the base of the tongue. Now, they're actually so big that they contain about half of all of our taste buds. Now, the foliate, think flowering foliate on the sides here. These foliate papillae are located on the lateral aspects of our tongue. And lastly, the fungiform papillae are located at the very tip of our tongue. Now, an interesting fact about the fungiform papillae is that they're completely translucent. So if there was no blood, we would actually be able to see right through them. But their blood flow actually makes them appear as red. Now, another interesting fact to know is that the back third of the tongue is actually innervated by cranial nerve 9. So if we look at the shape of 9, we can actually see it kind of looks like a little tongue and a throat. This will actually help you remember that it's called the glosso meaning tongue, pharyngeal meaning throat nerve. So we see it's the glossopharyngeal nerve. Now, the front or the anterior 2/3 of our tongue are actually innervated by our cranial nerve 7, our facial nerve. Now, if you must remember this for some sort of test or clinical, I don't know why they would be asking you this, but please remember that the fungiform taste papillae are innervated solely by a branch or an aspect of cranial nerve 7 called the chorda tympani. So now that we know some basic anatomy, we've cleared up some controversy about the tongue, we can actually discuss how we taste. So every aspect of our tongue, our entire tongue, can taste every one of the 5 primary aspects of taste, which are bitter, sour, salty, sweet, and savory. But it must be known that certain aspects of our tongue are more sensitive to these types of taste than others. So the base of our tongue is actually more sensitive to bitter taste. Remember, bitter in the back, bitter in the back. Now, the posterior lateral aspects, meaning the back sides of our tongue, are more sensitive to sour. Now, sour meaning like a lemon. Now, the anterior, the more forward lateral side aspects of our tongue are more sensitive to salty salty foods. This could be like a pretzel. Now, the tip of our tongue is more sensitive to savory and sweet taste. Now, savory is sometimes called umami. So remember, bitter in the back, sour, salty sides, and save the savory and sweets for the tip of the tongue. So let's say that we've drank something bitter, like coffee, black coffee, not a frappuccino. We're just talking about black, bitter coffee. Now, where would we sense this? Well, we know that bitter is in the back. So in the back of our tongue, we know that we're going to use cranial nerve 9. And then with our circumvallate papillae, with our little taste buds and taste receptors on them. So the bitter tastants actually use the phospholipase C IP3 protein kinase C pathway, which we know from our previous videos actually involves the increase in intracellular calcium, which will depolarize our afferent neurons, afferent meaning traveling up to our brain, which are connected to our taste receptors. So bitter at the base. Well, what about salty and sour? Well, these are a little bit different. So let's say that we ate some salted lemon. Where would we taste this? Well, we know we would taste the sour lemon and the salty lemon. We would taste these on the posterior and anterior lateral aspects of our tongue by using our foliate papillae and cranial nerve 7. These sour and salty tastants work in sort of similar ways. So they involve the influx of hydrogen for sour and sodium for saltiness, right? They involve the influx of these two cations to depolarize our taste receptors directly. So these are cations. We know they're going to enter the taste receptor. They're going to depolarize it directly. But how about sweet and savory, sweet and umami, like a maple bacon donut? Well, where would we taste it? We know that we would taste this, it would be more sensitive at the tip of our tongue using our translucent fungiform papillae while using the chorda tympani branch of the facial nerve, cranial nerve 7. These taste receptors also use the phospholipase C IP3 protein kinase C pathway to eventually depolarize the receptors and afferent nerves. So eventually, these afferent neurons, they are going to synapse in our primary taste cortex of our brain, where our brain can take the combination of these flavors and then turn them into an idea or an interpretation of what we are actually putting on our tongue. As always, thank you so much for taking the time to learn with us today, and I hope that you like and subscribe for more content.